Hi, and welcome back to The Cutting Edge with me, Shribos. It's been a rough week for the world, but even during this time, there are fascinating discoveries going on in the world of science. So let's celebrate some of those together. Now today, I want to tell you about some recently discovered and really cool links between cancer and bacteria. Now, you might know that there's bad bacteria, like the kinds that you wash off your hands to avoid getting sick, and good bacteria, like the trillions of bacteria that live in your gut, which make up your gut microbiome, which are really important for your health. And it turns out that tumors actually have their own microbiomes, bacteria inside cancer cells, which were the recent subject of a fascinating paper which came out in the journal Science this week. By studying over 1,000 tumors from patients, this study was able to show that different types of tumors, breast, pancreatic, brain, each have different bacteria. We don't exactly know which bacteria is doing what in tumors, but some of the initial things they saw are really cool. Like the fact that there's different bacteria in lung tumors from smokers versus people who have never touched a cigarette. And maybe there's a reason for that. You probably know that cigarette toxins can cause cancer, but those toxins can also mess with cancer cells. Now, at first that might sound great, like smoking can treat the cancer, but it turns out that the bacteria in the tumors from smokers are actually known to get rid of those toxins, maybe protecting the cells as they grow into a tumor. They also found that breast cancers had the most bacteria in general. Breast tumors, which have little sensors for the hormone estrogen, are also known to have lots of free radicals. Or if you think back to atomic structures, basically oxygen molecules which are missing one electron, making them super sticky and able to mess with basically everything else in the cell. And it turns out that those tumors also have bacteria, which make antioxidants, basically little molecules that can donate that missing electron and neutralize the stickiness, maybe letting cancer cells keep growing. Now, figuring out what bacteria are doing in tumors is going to take a lot more time and a lot more exploration. But what makes this paper groundbreaking is the way in which they systematically figured out which bacteria was in which tumor, with enough samples for those trends to actually mean something. So to talk about that, let's first talk about bacteria. Bacteria have different machinery than our cells, their own DNA, RNA, proteins. If you think of it sort of like the text which is illustrated into a picture and then 3D printed to make the actual thing, this is basically like red bacterial versions of all of it, including our little illustrator dude and the 3D printer. The ribosome, the 3D printer, is actually made up of a bunch of different pieces of ribosomal RNA, which if about now you're starting to ask, wait Shree, isn't RNA the blueprint picture? You would be absolutely right. Imagine this sort of like blueprints with markings on where they should be folded to make the origami of the 3D printer. Now, in bacteria, the 3D printer has this one super important origami piece that we can unfold and study called the 16S rRNA subunit. It's a chunk that's basically found in all bacteria, but have these variable regions, the colors that are different between different bacteria. But because a few copies maybe aren't enough to make sure you're seeing the colors of those 9 regions correctly, we have a tool called PCR, which actually lets us copy only those chunks a ton of times. And after that, by looking at the unique pattern of colors, we can figure out which bacteria is which. And that's what this paper did on a huge scale, giving us a massive set of data on which bacteria is found in which tumors. Now, finding out that tumor cells have bacteria inside them is in itself a really big deal. But the fact that some types of bacteria are always found in breast cancers or pancreatic cancers or lung cancers can be revolutionary. Maybe we can target tumor bacteria to make cancer cells more responsive to our drugs. Maybe that means we can use antibiotics to kill the bacteria and prevent tumors from forming. Maybe we make drugs that are fine everywhere else in the body, but are converted into poison specifically at the tumor by the bacteria? Those are all fascinating questions which will hopefully be explored in the coming years. But until then, I hope you join me next time as we dive into even more cutting-edge science.